Hello, students. Today we're going to talk about amino acids, and this is chapter 3. We have to have a really great understanding of their properties. We're going to see that some of them are acidic, basic, polar, nonpolar, and their properties are going to affect their role and position within a protein. So chapter 4 deals with proteins, but we're going to study a lot of different kinds of proteins in the semester. And one of the common themes as we progress to that is what are the key amino acids that are involved in that protein, and how are their properties involved in their function. So proteins are made up of alpha amino acids, and they will have some similarities to each other, and then they'll have differences that make them unique amino acids. So our main structure for amino acids will have this alpha carbon here, and then it'll have a carboxylate group up here, an amino group here, hydrogen, and then an important R group that gives it its distinctive properties, almost like its characteristic, its character. And so a couple comments is that if you prefer, you can write some of these structures out in line form. I will always be writing them as out like this, just because I think for teaching purposes, students can see more what's happening. But if you prefer the line structures, that's fine. And you're going to see them here coming up in a second. Okay, one of the first things that we need to address on amino acids is that most of them have what's called chirality. So for molecules to be chiral molecules have 4 different groups around that alpha carbon. And the 4 different groups are going to be assigned a priority depending upon what the groups are. So in this case, the amino group is 1, the carboxylate is 2, the R group is 3, and the hydrogen is 4. And then recall that you can determine its stereoisomerism based on the direction of counting these groups. So notice how when I count in this one, I'm going in a counterclockwise position. That would be the S chiral configuration. And then there's the R, and S and R differ from each other as non-superimposable mirror images. So a chiral molecule will have a mirror image that you cannot superimpose on it. Now, there's also another type of designation, which is the L&D designation. That's based on an absolute configuration where the standard is glyceraldehyde. And so that has a different way of looking at stereochemistry by comparing these isomers to a standard. So in amino acids, the L isomer is what's incorporated into proteins. Okay, so let me just stop here for 1 second. All of what I said was probably a full chapter in your OChem. So I don't want to go back through and discuss it in that depth. So if you're not remembering R and S and L and D, go ahead and, try to think about that from your previous course. I don't specifically test you on that part. So I'm not going to ask you to determine whether something's R and S or L and D. I just want you to try and remember that chirality is important in molecules that have 4 different groups around the carbon, and that in the case of biochemical molecules, it's very common that one isomer is preferred over the other. So in proteins, L is preferred. Okay, so that's a little bit different than your OChem class that gave you like here's the following molecules. Determine if it's R and S, R versus S, or L versus D. Okay, we don't need to do that part. Okay, but what we'll see as we move forward is that chirality does play a huge role in biochemical molecules. I'm going to skip this slide. We'll come back to these in one form or another later. Okay, so common amino acids and their classifications. Couple things. Amino acids have a 3-letter designation and a 1-letter designation. So you should know their 3-letter designation and 1 letter for all of them. Okay, so here's the deal, is that on your test, if you don't know them, it's just going to take you too long to kind of know what you're looking at and what I'm referencing. So there is incentive then to learn these designations, even though we're online and you'd have your reference materials in front of you. So every time I give you a letter designation as part of a multiple choice, let's say, if you don't know it, you're going to spend way too much time looking up information. Not to mention that this biochemistry class is a prereq for other courses that you have to take. So they are going to expect you to know certain things. So I don't think this is too much to ask. Now, depending on your future goals, you may want to invest in learning all 20 amino acids. I think for us, I'm going to highlight as we go ones that I think you should know and memorize, just to kind of have under your belt as we move forward. So you do not have to memorize all their structures, but there's some that come up for us more than others. Okay, so let's begin. At the top, there's these non-polar aliphatic amino acids, meaning carbon-chained amino acids. And so, as you know from hydrocarbons are non-polar. So these for sure would be hydrophobic. Now, I know we haven't talked proteins yet, but what I want you to think about is that it is unlikely that these amino acids would be in direct contact with water. Instead, these types of amino acids would be favored buried deep within a protein structure, away from the outside water. So that's kind of where we're headed in understanding how their structure affects their properties and their locations within a protein. Okay, so glycine is just an H. So, it's not really these hydrocarbon ones. I kind of see it as it's also non-polar in the sense that it doesn't have a polarity to it. It's also not chiral. Why isn't it chiral? Well, it's not chiral because it doesn't have 4 different groups around that alpha carbon. So the alpha carbon is just implied here. So that one's not chiral. The rest of them are, because I'll have 4 different substituents around alpha carbon. The one nice thing I like about this slide is that it gives us the 3D shapes to the amino acids. Alpha carbon will have a tetrahedral shape, and so it's drawn that way. Later, when I start drawing out some things, I'm not going to include that geometry, but I do think it's important to note that they do have a 3-dimensional shape to them. Okay, out of this bunch, I would say for sure, know your glycine. That one's easy. I'll have alanine, not necessarily a critical amino acid, but just gives you an example of a aliphatic one with a hydrocarbon. Okay, these are all hydrophobic and nonpolar. Next, proline. Proline is very unique. I won't have you memorize its structure, but I would say be able to recognize it. And it's a little bit easier to recognize, I think, because it has an intramolecular bond. You see how this is alpha carbon, and then it has the NH group, but then you see how it's a ring that goes all the way around. So this one here is the only one that doesn't have an NH3 plus group. And that's because it has this intramolecular bond and ring structure. Later, we're going to see that proline is very unique, and it's so unique that it's not going to be able to be found everywhere in a protein because it has sort of this, it creates kind of a kink in this chain. And we'll also see that it's a dominant amino acid in a protein called collagen. We'll come back to that in chapter 4. Then you have methionine, okay, also nonpolar. And then we have the nonpolar aromatic amino acids. If you recall from your OChem, the aromatic rings, right? Aromaticity. And so there, phenylalanine has this big bulky ring, tyrosine, tryptophan. Notice from these guys that they are very bulky groups. And so again, it's going to be kind of limited where they might be found in an overall protein structure. If you had because it's like space, right? They're going to have enough room for these big bulky groups. Now is a good time to mention that their letter designations don't always match the first letter. So P got used up with proline, so then they went to F for phenylalanine. Tyrosine is Y, and tryptophan is W. Okay. Okay, our polar amino acids, important ones for you guys to memorize. For sure, memorize serine and cysteine. Okay, our polar amino acids have polar groups. So if you recall from our previous discussion in polarity, OH hydrogen bonding, OH hydrogen bonding, NH2 hydrogen bonding group, NH2. SH is also polar, but that's just a dipole-dipole because it doesn't fall in the category of hydrogen bonding. Okay, remember hydrogen bonding, OH, NH, or FH. Okay, and so these guys would be definitely hydrophilic. And if we're talking about later amino acids that can interact with water, these would be highly interactive with water. Okay. Other polar amino acids are these positively charged amino acids. Lysine, you should know its structure. Okay, from memory. Histidine, I would say for sure be able to recognize. Okay, and then you have these negatively charged amino acids like aspartic acid. Okay, that's also one I'd like you to know from memory. Okay, these essentially have charge to them, and so they would be able to do an ion to water interaction. Okay, and then also, if you think about it, if let's say aspartic acid and glutamic acid, like they won't be attracted to each other because they're the same charge. So within a protein, if you're going to have aspartic acid, more than likely you would have one of the positively charged amino acids near it to stabilize it. Okay, so in terms of the aliphatic amino acids, proline, alanine, valine, leucine, isoleucine, the more carbons it has, the more hydrophobic it is. So that makes sense, right? The bulkier the carbon chain, the more carbons, the more hydrophobic it would be. And all right. The aromatic rings are very, very bulky. Phenylalanine there, just a CH2 with the benzene ring, aromatic ring. Tyrosine has an ionizable hydroxyl group, and we're going to talk more about that a little bit later. And then to use some organic chem language, the tryptophan has this indole ring, bicyclic indole ring. Okay, so this particular slide is just going to show you that some properties that might be unique to these aromatic rings are that they will absorb ultraviolet light at 280 nanometers. And so anyways, it's more an FYI of another property, absorption of light. And we have machines, and I'll just go back to the previous slide, that are called spectrophotometers, that you shine light at a certain wavelength, and a solution will absorb some of the light and then let the rest of it transmit. So in the case of tyrosine and phenylalanine, they absorb UV light at 280 nanometers, and that can be then allows us to do a quantitative description, determine how much of those amino acids are in a sample. Now, if you're wondering, would I ask this slide? No, this is just to give you an impression of different properties that they have depending upon their structure. Okay, methionine is the starting amino acid for almost all proteins. That has to do with the coding on the DNA that codes for proteins. We get to that at the end of the semester. It's very hydrophobic. Cysteine is one that I think you really need to know. It has this CH2SH group. It's going to be able to form very interesting linkages. So this is showing that linkage. So here are two cysteines, and what they're going to do is there's going to be a covalent bond that's formed between the two S's, and this is what's called a disulfide linkage. And these, in the process, are going to lose their H, right? You see these H's here? So that's an oxidation reaction, the loss of hydrogen. Conversely, the other direction is the gain of hydrogen, and that's a reduction. One of the interesting things is that in your hair, the cysteine, the disulfide linkages kind of help to create different patterns in our hair. So some of us have straighter hair than others, more curly hair. And all the hair products that you might use that involve either curling your hair like a perm or straightening your hair involve disrupting and manipulating these disulfide linkages. So I was your age in the 80s, and in the 80s, perming was a big thing. So what a perm does is it takes your hair that has these disulfide linkages here, it adds a chemical to reduce them. So you come to this side over here, and then they tie your hair around a big curler, and then they would add a chemical to go back to reforming the disulfide linkages, and then your hair would have a wavier pattern, a different pattern based on how it was rolled into those rollers. So this is a great example of disulfide linkages. Straightening process would be the reverse of that, okay, which I think is more your generation to have used chemical straighteners. Yes. Okay. So cysteine, I just noticed, is misspelled on the slide a little bit. So the correct spelling is here. Okay. Acidic amino acids. So aspartic acid here's aspartic acid. Okay. It's a carboxylic acid. And so it's going to be named like one. So aspartic acid with the IC ending. Now, if it's ionized, its ion is shown on this graphic. It's lost its H plus, the ending goes to ATE. So I just want to show you that because most textbooks assume you can figure that out and they don't tell you that, and it can be very confusing. The other thing to note is that there's sort of a, I would say on the internet in particular, not in your book, sort of a messiness that comes with these. So in other words, sometimes they'll write it as the acid, but they'll call it aspartate. Aspartate is really the name when it's lost its H plus. So just know that so it's not confusing if you see it incorrectly somewhere, right? And that you know that they're essentially talking about the same amino acid, one that's protonated and the other one that's deprotonated. Okay, same thing with glutamate. Deprotonated glutamic acid two CH2s would have the COOH, the H on it. Okay. Now, in a minute, we're going to talk about which one of these exists at pH 7.4, and it is considered an acidic. These are both acids, acidic amino acids. But we're going to look more closely at why we write them a certain way and how that's pH dependent. Our basic amino acid, the most famous one for sure is histidine. And I asked you to recognize it. You don't have to write it, but I asked you to recognize it. And you see here the basic group. Oops, I wanted to get back to my pen. When that H is deprotonated, it has that basic nitrogen group. And then remember, nitrogen with 4 bonds has a plus charge. So N with 4 bonds goes to a plus formal charge. Okay, and this is showing that as sort of going back and forth depending on the pH, and we'll get to that in just a moment. Okay, histidine is a super important amino acid. So, it's one that we're going to see over and over again in different proteins. Lysine is a diamino acid, which means that if you look at it closely, it has an NH3 group on the R group in addition to this amino group. So 2 amino groups. And then arginine also is positively charged. Okay, R group. And we're going to talk about understanding why we would draw it as NH3 plus in just a moment. Okay. Okay, serine looks like alanine, but with a hydroxyl group. Super important amino acid that plays a big role in the active site of enzymes. Also threonine as well, also important in the active site of enzymes. So we will kind of see that. That'll be later in, I believe, chapter 7. And then here's asparagine and glutamine. Okay, so I'm not going to read everything on this slide, but the main idea, and I already said what's on this slide, is if amino acids are nonpolar in a protein, they're going to be buried in the interior of the protein, getting away from water. Hydrophilic ones are going to be on the exterior of the protein and in contact with water. This also talks about the importance of histidine. And then you see the fourth bullet point, hydroxyl and thiol groups can function as nucleophiles. That's our cysteine. Our serine. Oh yeah, cysteine and serine. But I want you to know that we are going to talk more and more about this. And this particular slide doesn't leave us the entire semester. So instead of memorizing the slide, it's important for us to be able to look at the amino acid and know what kinds of properties they have. Okay, now let's talk about how we're going to predict the charges of these side chains. This is a slide that you have here. I actually prefer the one from your textbook. So the one from your textbook is here. And this is one that I shared with you if you were at the Zoom lecture through the file that said amino acids for exam. And it's also posted on the Canvas. This is your new periodic table. So this is a reference material that you will have with you. You're not memorizing. Okay, but let's take a look at it. Basically, if we have an amino acid, so I'll draw it again here. See this alpha COOH? That's this one. And these are listed here as your pKa values. Now, I just said to you guys that you don't need to memorize these, but let's look at the numbers. If you notice, every single one is around a pKa of 2. Those of you that are using the course as a kind of a review for some sort of test of some sort, I would actually say you should for sure memorize that they're all around 2. I think it's just helpful to know that. But I also want you to realize that I will be giving you this for their exact numbers. For the alpha amino group here, those ones are at around a pKa of 9, right? Between 9 and 10, okay? And then the R group, most of them do not have an R group that has any acidic properties. But down at the bottom, we do have a group where the R group has acidic properties. Now, they're not all the same. As we've talked about before, the lower the pKa, the more acidic. So these first 3 really seem like weak acids, but look up here, like arginine at a pKa of 12.48. On paper, we could say that has acidic properties, but really that is a very high pKa, which means that it is very, very weakly acidic. Okay. All right, so I'm gonna ask you to either A, print this, B, as you're watching this, have your open up the amino acids for exams, this is on there, or have your book out. So your textbook has this. And I actually believe that your textbook's really great in this particular chapter. I think it's a really good reference. So it has some great pictures of the amino acid. It explains them very well. So as we move forward, I would really need you to have this information at your fingertips. So pause me if you need to grab it. Okay, so with that in mind, I'm going to grab my reference. Hold on, pause me. Okay, so what I'm going to be doing now is let's start with glycine, our easiest amino acid. And we're going to look up the pKa values for glycine on our chart. So for glycine, this one is 2.34. And the amino one is 9.60. Okay, at the same time, I'm going to draw a little titration curve because we're going to draw it as we go along. Okay, so we're going to start here at a low pH. And we're going to picture that we're going to titrate or raise the pH. And as the pH rises, we're eventually going to deprotonate the carboxylic acid first because it has a lower pKa. It's more acidic, so it's going to go first. Okay, and that pKa is 2.34. Now on my chart, it's going to look like this. And we're going to start off with glycine. I'm going to change the color on it. Structure A. I'm going to start off with structure A. And then we're going to end up at structure B. That's going to be, see how this is acid and conjugate base? We're going to see only that's conjugate base at first equivalence. Now, as we've been discussing and learning, at halfway to first equivalence, we have that spot where we have 50%, I'm going to just put pH equals pKa, plus the log of structure B over structure A. And at that point here, where I put the X, you're going to have equal amounts of A and B, 50-50 in the mixture. And at that special point, we're going to have a pH that equals pKa. Okay? We keep adding base, ultimately, the amino acid is going to deprotonate at the amino group. That's going to happen at a much higher pH to get a structure C. So our second equivalence point will have only structure C. Halfway there, you're going to have a structure, right? You're going to have pH equals pKa plus the log of structure C over structure B. That's going to have 50-50 of each. And at that point, your pH is going to equal pKa. But the second one, the 9.60. Okay, so the important point here is we can use these titration curves to figure out the pKa values of our amino acids. The other important part is that really will dominate our discussions will be, what is the structure? At pH 7.4. And so what's the structure at pH 7.4? Now my graph isn't so pretty. There's a prettier one coming up later of this exact one. But if we look closely, we would see that 7.4 would be somewhere like around in here, where I've highlighted in yellow. Okay, somewhere in there. And so the idea is as follows, is that more than likely 7.4 is somewhere in here. And so what structure is going to dominate? It won't be the only structure, right? But it's what's one's going to dominate is going to be structure B. So structure B is what's going to dominate at pH 7.4. Because you see how at the first equivalence, there's only B, but we're past it. So it won't be only B, there'll be some C in there, right? But what's going to dominate is structure B. And that's why anytime you draw glycine at pH 7.4, it should look like structure B. Now, there's a whole lot of things on the internet that are wrong on this. And so I highly recommend that you just look at your book. Because if you Google, like, what's the structure of glycine, some references will show structure C, some A, some B. And it's just better to understand why it has the structure it does based on its pKa's. One last thing that's used a lot experimentally. That's used a lot experimentally is what's called the PI, the isoelectric point. PI is the pH where you only have the neutral amino acid. And that's called, the neutral one is called zwitterion. So in our case, this in glycine, the structure B is the zwitterion because it's neutral. You see how it's a plus and a minus. So at pH 7.4, it's a zwitterion. Now, where on the titration curve do we have only B? That would be first equivalence. So how are we going to figure out the pH at this point? Okay, and the way we can do that is, if you notice, it's the midpoint between the two pKa's. So I would take 9.60 plus 2.34 divided by 2, and that should give me 5.97. That is the PI, isoelectric pH. Okay, so let's talk about another one. Let's do glutamic acid, or let's do aspartic acid. Make sure that I don't have it on highlighter. So this is aspartic acid, or aspartate. I'm going to draw it initially as fully protonated. try to get rid of that weird N that I drew. Okay, so go ahead and let's look up the pKa's because you have your reference material. ASP 2.09. This one's 9.82. And then the R group is 3.86. Okay, so the pKa's change a little bit depending on the structure of the molecule. All right, so let's draw our titration curve. And then I'm going to grab a different colored pen. We'll call this structure A, and we'll start with A. Now it has 3 ionizable groups, so it's going to have 1, 2, 3. Okay. All right, so I'm starting off with A, and then the first one that's going to go is the COOH that's attached to alpha carbon. So that's our first one to be ionized. So let's go ahead and write that. Okay. All right, so that structure A has now gone into what I would call structure B. And then where do we have only B is in the first steeply rising part, our first equivalence. And then 50-50 point, halfway there, that's our first pKa at 2.09. So that's 2.09. Okay. All right, so that's structure B. If we look at its charge, that's our Zwitter ion. Okay, see how it's neutral? All right, so now we're going to look at the pKa's and see who ionizes next, and that would be the H on the R group, right? Because it's at 3.86, so that guy's up next for deprotonation as we increase the pH. Okay, and then the last one to deprotonate is the amino group. Okay, so now we're going to have structure C and structure D. Okay, so we have first equivalence, we'll have only B. Second equivalence, we'll have only C. And third equivalence, we'll have only D. And then we can highlight, and maybe I should have put this in a different color. I'll use blue. So here's the first pKa. Here's the second one. That would be at 3.86. The next one would be at 9.82. Okay, now your book has prettier pictures of these and more accurate. Sometimes their graphs aren't so, you'll notice it in your textbook, they're not so perfectly like flat in the pKa area. Okay, and remember this is pH on this side and volume of hydroxide over here. All right, now let's take a look and analyze this. where would pH 7.4 be? It's going to be somewhere in this area. I'm going to do yellow highlighter. Somewhere in here, you know, somewhere around here. I don't have this well spaced out, but so for that, then the structure that we can see will dominate at 7.4 has to be the structure C. So this is the structure that's going to dominate at pH 7.4 because it's in between 3.8, 7.4, somewhere in between 3.86 and 9.82, meaning that the R group is deprotonated. So anytime that we want to show aspartic acid as an amino acid, it's really deprotonated, and that's why normally we call it aspartate ion. It's also negatively charged. R group. Okay, lastly, for the PI, we have to really think about this guy. At pH 7.4, this amino acid is not zwitterion. The zwitterion is structure B, right? You see that? This was zwitterion. So the PI then is we have to kind of think about where is their structure B only. That's at the first equivalence point. So the PI is going to be the average between 3.86 and 2.09. So let me average those. So I got 2.98. Okay, so when they have an R group that has a pKa, the zwitterion, the PI, you have to look at closely to see where the zwitterion structure is in relation to the pKas. Okay, so this is a big part of your first exam, is understanding these amino acids. And so what I would like you to do is to take a look at worksheet number 2, which deals with amino acids and their titration curves. Okay, this guy is due on, I switched the due date. So I believe, hold up, that I switched it to September 10th. Now, what I'm really hoping is that you have attempted this worksheet so that we can go over it in Zoom on September 8th. And this is where we're really going to dive into all this and make sure that you understand it, because this is a huge part of your first exam. Okay, and all right. Here's a prettier picture of the one for glycine. And so there was, we already calculated the PI at 5.97. That's the first equivalence point. There's pKa1. So we did this one. This just is a nicer graphic than my graph. Here's glutamate. So I'm going to do this. I'm going to, whoops, you're looking at it. You shouldn't look at it. Okay. So try glutamate. Okay. So draw the structure out, take the pKas, draw the structures at different as it deprotonates, and then draw the titration curve, label the pKa's, and find PI, and then you can check your answer on the next slide. So that's what's going to be really nice about doing it that way. So pause me and do glutamate as an example. So for glutamate, explaining the example, the first pKa was 2.19. That's that COOH there, so that's gone first. The second pKa is 4.25. That's the carboxylic acid on the R group, because here's your R group. And then lastly, you lose the NH3 plus, H plus. So first pKa one is 2.19. The second one is 4.25, and this one's 9.67. Do you notice though here, I know that the inflection point isn't so strong. That's an experimental observation with glutamate. The titration curve's not quite as dramatic on that first equivalence point. Okay, now on a test, I find that very confusing. So I would draw them, and I tend to give you examples where you can see first equivalence, see how this one's like a little bit tougher to view. Okay, and then which structure, so let's draw A, B, C, and D. So we start off with just A. At first equivalence, we have only B. At second equivalence, we have only C. And D's up here somewhere. Okay. All right. So at pH 7.4, here's 7 in between here, you really are looking at, go across, structure C. Okay, structure C is the dominant structure for glutamic acid, but we'd really call it glutamate because it's deprotonated R group. Where's the Zwitter ion? The Zwitter ion is structure B. And for Zwitter ion, where do we have structure B only? Is at first equivalence. And that would be then where we have the PI, and that would be the average between 4.25 and 2.19. So 3.22. Okay. All right, so work on worksheet 2 for really great practice, and then we're going to be going over that next time I see you. I am going to stop here for this video lecture, give you an opportunity to start learning those amino acids. I will be continuing, though, talking about peptide linkages in the next video lecture.